Fine.
I have to read a policy. Oh no, apparently I've read it now instantaneously. So recording the transit. Perfect. Stop recording. Yeah, so I will put the recording onto the YouTube channel and then on the iBand website and the MST and all that kind of stuff. Probably tomorrow. It's actually doing a transcript of my voice, which is, I don't want that at all. Let's stop the transcript. MS Teams changes constantly. It should be happy. So, Michelle, I'll pass on to you first before Karen starts, and then I can introduce Karen.
Hi, just a short announcement. So before we have today's presentation, I want to invite you for the next meeting, December 15. So it's also Wednesday in 3 weeks. This will be a last meeting of this year. So obviously we'll make some summary, what happened in 2021, probably some good stuff, not only related to COVID. And we will talk about plans for 2022. So the events that we plan, and also we would like to have some open sessions. So no any particular presentation is planned for that meeting, but plenty time to chat and to discuss what to do and how to satisfy your needs better in next year. So we invite you to meet us in mid of December. See you there.
Thanks, Richard. What I'll do is I'll send around the MS Teams invite through MS Teams, but also send a link via email because I have no idea how savvy different groups are with regards to Teams. But it looks like everyone's here, and as far as I know, no one's emailed me in panic, which is a good thing, I guess. So anyway, well, welcome everyone. I might as well start. I don't know what numbers we've got here. We've got well, I've got 40 people. That's good. It's not bad for late on a Wednesday. It's Wine Wednesday, of course. So I'd like to introduce Karen Heeter. If you don't know her already, she's a PhD candidate at the University of Idaho and has been well, basically her lockdown PhD mostly has been playing around with blue intensity across much of North America, from the deep Southwest right up to the Northeast and the Northwest. Her talk today is going to be on aspects of that work and some of the things she's learned about, some of the things that she may have issues with, I don't know. Just a bit of background. I was intrigued about her background, but she has a BSc degree in ethnobotany, and then that was followed by an MSc in ecology and conservation biology, both I assume at Frostburg State University in Maryland. Her interests fall within the domains of biogeography, plant ecology, and paleoclimatology. If she's not coring trees, and I honestly don't know how, she must have been the only dendrochronologist actually doing fieldwork for the last 18 months. It seems that a lot of North Americans I know didn't do anything. But anyway, when she's not coring trees, she enjoys doing botanical illustrations. So if you need any books, if you're writing a book and need some illustrations, this is your go-to person, I guess. She plays the cello. And I'd love to see her and rides motorbikes. So riding the motorbike with the cello on her back would be probably quite interesting. But so anyway, that's Karen's brief background. I've been following her work. Looks good. I've been involved a little bit. So hopefully you can share your screen. Yeah, I would advise everyone maybe to switch the videos off just to keep the bandwidth stable. Keep your microphones off for now, and then at the end we'll open it up for questions when Karen is finished. So thanks very much, and hopefully this works, Karen. Yep, brilliant.
See all seeing this okay?
Yep, that looks good.
Great. All right. Well, thank you, Rob, for the introduction. As he said, this was kind of a great invitation to be able to talk about my my dissertation work over the last 3 years. And so I will say I was extremely lucky, partially I think because of proximity in northern Idaho, of the ability to do field work. So that being said, I'm going to give you guys kind of just a taste of what I've been working on across the United States with blue intensity methods. So just kind of a brief outline. I want to start for those of you that maybe don't use blue intensity for paleoclimatology purposes, or are just new to blue intensity, to kind of just give an introduction about temperature proxies from tree rings, particularly in North America, kind of what's been done and where we're going, and then my interpretation of blue intensity itself. And then after that, I'll get into kind of a pilot study where my journey on blue intensity began back in 2018 in the eastern United States. And then kind of how that spurred into the bulk of my dissertation work, which will be related to reconstructing temperatures with blue intensity across the western United States. And so, of course, that'll kind of be my methods results discussion. And then lastly, I'll follow up with some current and future work for North America and kind of where I see myself going next. So kind of to start, if we situate ourselves within the broader conversation of looking at climate change, right, paleo data is critical for understanding the long-term context to these modern trends, especially with temperature. And so depending on the resolution of our question, that can depend on which type of proxy record we use. So for terrestrial proxies, tree rings are critically important because they allow us to have accurately dated proxy records at the annual resolution. And so this is pretty unique. And of course, our mode of getting these proxy data are derived from this principle of limiting factor for tree growth. And so for those of you that maybe aren't super aware of that, what that means, it's this idea that tree growth, whether it's radial or densitometric growth, is dominantly driven by some sort of environmental factor that is the stressor, if you will. So in many cases, especially in the western US, this is oftentimes moisture availability, but it can also be temperature. Thus, we can get these temperature proxies from tree rings. And so where this has historically been done is at high latitudes or at high elevations in the Northern Hemisphere. And so these are predominantly derived from conifers. And then, of course, the tree ring metrics that we can use to develop these proxy records. We've got ring width, so whether it's late wood, early wood, or full ring width, as well as representations of ring density. So the maximum late wood density, which I'll abbreviate for the rest of this talk as MXD, and then now blue intensity. And so then if we situate ourselves spatially within the Northern Hemisphere, I wanted to start by kind of seeing like what has been done, especially for North America, where I'm currently working. And so I wanted to start by looking at the work by Briffa and Schweingruber throughout the 1980s and 1990s. This tremendous effort to develop a network of MXD records across the Northern Hemisphere. Particularly, I'm interested in what I've circled here in red, the Western United States. Historically, we think about where temperature proxies exist, and they for a long time were limited to the high latitudes in North America. And partially, this is because, tree ring studies it's not really that old of a field. And you think about, we've been working with radial growth metrics for a long time. And in places like the arid Southwest, ring width just isn't always the best predictor of temperature because that hydroclimate signal is competing for that dominant limiting factor, I suppose. And so MXD kind of was a bit of a game changer when it came to, okay, well, can we look to the high elevation locations in the lower latitudes to derive these temperature proxies? And so that's really where it began for North America. And then fast forward to 2016, where we have the N-Trend network, which is, again, a huge synthesis of MXD, ring width, and blue intensity records for North America. And this is great, but that being said, there are still spatial gaps that exist. When we think about the end trend network, for example, not including any records below 40 degrees latitude. And again, in part, that's probably because of that complex hydroclimate interaction with tree ring records that far south. And so kind of had me kind of thinking about, so where are the research needs spatially? And so you see, for at least the Northern Hemisphere, North America is a pretty large spatial gap. So, and then thinking about, okay, well, we've got these great MXD records. So how do we make the jump between X-ray based techniques to visible light techniques, especially for the purposes of driving these temperature proxy records? MXD is shown to work really, really well for reconstructing summertime temperatures, especially in North America, but it's pretty cost intensive as well as time intensive. And so for that reason, or partially for that reason, a lot of these Briffa-Schweingruber sites have not been updated. And so if we want to contextualize the most modern trends, as well as be able to calibrate our models to include the last 30 years, it's kind of the ability to go and update all these MXD records across the United States just wasn't really feasible for me in my lab. So I got interested in this idea of visible light techniques. And so kind of to show a visible example here is when we think about marker rings with ring width, we're usually looking for a narrow ring that allows us to anchor these dates in time. And sometimes we can actually see that visibly with density as well. And so this is an example I'll use. That's an Engelmann spruce from British Columbia. They don't always work out this well, but this one, you can notice that sometimes these rings appear lighter than maybe the rings around it. So 1954 is what I'm using as my example. And so that kind of, when you think about that jump between how do we go from MXD to blue intensity, it's not exactly linear in my mind, but sometimes, if you think about blue intensity and MXD both being representative metrics of density, it's kind of, to me, where you can kind of see this start. So visibly, it looks different than the rings around it, as in the late wood might be lighter in color, the cell walls might be thinner, that sort of thing. And so where we really start to see this switch towards visible light is the work by Shepard et al. They ran brightness values against late wood density, a population of red spruce up in Maine. And so they compare late wood brightness and late wood density together and then against temperature, and they see that they actually both do correlate with April through May average temperatures. So this is kind of the beginning of that examination of, if MXD is cost intensive and time intensive, is there a new way of looking at this representation of density that might be a little bit maybe more accessible to different labs? And so this schematic is something I put together to kind of just break down what we're actually looking at in modern times with blue intensity. So in panel A, if we start with just the visible light spectrum, we are interested in just that blue wavelength. And so this blue intensity, of course, as I'm sure most of you are aware, can be collected using just a commercial flatbed scanner. It's a lot cheaper to work with. So then when we think about a scanned image of a core in panel B, of what that actually looks like in full color. But of course, scanners are additive color, and so it's an RGB kind of algorithm. And so what that translates to actually of what we're measuring is just that blue reflected light. And so I use a program called coRecorder. I'm sure, again, most of you are aware or use it readily. So not only does it allow you to delineate ring boundaries, but also to collect these different color channels for whatever window setting you're looking at. And so a lot of what I've been doing too is kind of playing around in coRecorder of different parameter window settings to see, okay, well, if I increase my depth or I increase my width or percent collected, how will that influence my overall signal? So I'll talk more about kind of these exploratory methods in just a minute. And so ultimately what you end up with in panel D is a linear time series like you would just with tree ring width. It's a measure of 0 to 255. But that being said, you're also able to look at the red and green channels, which has been kind of some fun exploratory stuff for me as well. But you even notice from the Engelmann spruce chronology I'm using, just as an example here, the variance is definitely better in the blue intensity or in the blue channel. And so to break down, what I've been looking at primarily has been latewood blue intensity. So of course, just the reflected blue light in the latewood portion. And so latewood blue intensity has been widely shown to be negatively correlated with MXD, wood density. And so if you invert it, of course, it behaves similarly in terms of climate response. So the idea that the Swanesea group kind of put forward is McCarroll and others, that it is a cost-effective and less time-intensive parameter to use that is similar to MXD. And so for my purposes of, I'm doing a 3-year field campaign for my dissertation, it definitely seemed like this was the way to go moving forward. And so just to kind of hone this in, I wanted to use a visual example from that Engelmann spruce I spoke about a few slides earlier, specifically for dendroclimatology, kind of relating BI with climate data. And so from the example I used a few slides ago, 1954 to me was the marker ring that seemed a little bit lighter in color than comparatively the rings around it. Inversely, you can look as you go down or go in time, 1958 seemed like maybe it was darker than rings around it, just visually speaking. I have not run any data on this yet. So I wanted to see, well, how does that kind of corroborate with the data? What's the relationship there to the instrumental climate data? And so on the left, here's an image from Q-Recorder that is the latewood blue intensity collected from the scans, and of course my marker rings that I've included as well, 1958, 1954. And so what I did is I pulled the CRU, just local instrumental data. I've got summertime, June through August mean temperatures on top in red, and then mean annual temperatures in black on the bottom. And so seeing actually, if you run the data and kind of look at the relationship here, 1954 is actually, for summertime temperatures, the coolest year on record for the instrumental period. Then similarly, 1958, actually until 2020, then 2021 was the warmest year on record for this area. So kind of for me making this connection between, we still do sometimes see these visual markers and they are correlated especially well with summertime temperatures. So where kind of it started for me then in terms of actually trying to put together some exploratory studies with blue intensity was in the eastern United States. And so this was a pilot study I put together in 2019. So as I mentioned, the Shepard et al. Work, where to me blue intensity in North America really began, was up in Maine, so just for spatial reference here. And so what I was interested in is kind of looking at, how do we get increased coverage at the mid to low latitudes of North America. So I started with a population of red spruce in North Carolina at 36 degrees latitude, so just that spatial reference there. So what I ended up doing is running delta blue intensity as well as latewood blue intensity, kind of comparing the two, and it turned out that that delta blue intensity metric is the one that I ended up going with. We had some discoloration issues that we had to get over. So then I ran it against the instrumental temperature data, both T-mean and T-max, and I found really promising results with maximum temperatures in the months of September and October. And so just to kind of translate this figure on the very far right, this climate response, for those of you that are not familiar, this is a running windowed correlation. So on the x-axis, it's just 10-year increments moving through time. And it's a Pearson correlation coefficient. So positive, strong correlations are in brown, and then the negative ones are in blue. And so we see strongest positive correlations through time that are somewhat temporally stable for September and October. But then on the bottom panel, looking at the ring width versus maximum temperature, we really don't see very strong or time-stable relationships with temperature at all. So then looking at the spatiality of these correlations with temperature over this full instrumental period, again, it's a Pearson's correlation coefficient, so the dark red being where it's the strongest, over the Appalachian Mountain Range. So spatially, this correlation makes sense as well. And so this was pretty promising in terms of looking at not only a new species with blue intensity, but also kind of getting at a lower latitude for these temperature records. To date, there really weren't any temperature proxy records for the Southeast United States, and so this is very good news, and I guess spearheaded this long field season throughout the southern Appalachian Mountains, targeting populations of red spruce and eastern hemlock. And I'm sure you're probably wondering, well, 6,500 feet is not particularly high elevation, but for the Appalachians, it's pretty high. Anything over 2,000 meters, we were happy with. And kind of what that turned into was an expansion upon that work that my advisor and I put together, looking at multiple populations of red spruce in eastern hemlock, just the climate response with the latewood blue intensity or the delta blue intensity, whichever the case was. And overall, we're seeing, pretty good climate response with these southernmost populations for red spruce. And so this was kind of pretty interesting, and definitely there's a lot more work to be done in this part of the country. But with that said, this was really good proof of concept, but, I was at University of Idaho, and then, things become uncertain, right? Especially now in hindsight with COVID. I kind of started to think, well, it might be a better use of my time to explore local research in the Western US and kind of see if I could take these methods elsewhere. And so that's exactly what I did for my dissertation. I ended up kind of going westward, and so I put the schematic together, this image on the right, kind of thinking about a checklist of things I would need to consider if I were going to develop a blue intensity study for the Western US. So I know that I'm interested in looking at sampling below 50 degrees north. So that would mean that I would need high elevation locations. So I would be working primarily in the subalpine belt. So of course, the Western US, we've got the Rocky Mountains, we've got the Cascades, and we've got the Sierra. So check that off the list. And in terms of species, right, I needed something that was a light color, it was consistent wood color, that you don't have, issues of overly resinous species, this heartwood sapwood discoloration. We're working on kind of as a community addressing those color bias issues. So then looking out, yeah, we do have a minimal species, but then also for the Western US, avoiding overly damaged trees. I think Jesper was talking about this last time. If you have, issues like reaction wood or scarred tissue, you're not truly getting that, clearest representation of density. So avoiding trees that were damaged, fire scarred, or insect outbreak, which if you are familiar with the ecologies of the Western US, those are two huge issues when trying to find trees. So ultimately, it seemed pretty promising, so I proceeded. So for this, I guess, next part of the talk, I'll kind of just discuss my sampling across the Western US and some of the trials, tribulations, and everything in between that comes with the territory of that. So methods. I cored a lot of trees. We did, I think it was 4 field campaigns across the Western US. Sampled a lot, got to involve students, which was great. Also got to probably go to some of the places that were bucket list places for me. This is Crater Lake, for example. So it was such a joy. But in doing that, we cored a bunch of different species that were new for blue intensity methods. And so kind of some of the issues that we had to deal with, of course, were the issues of heartwood sapwood transitions with color. And so the ones that I was looking at, this was predominantly an issue in anything in the Pseudotsuga, the Tsuga, Pinus, and Larix genuses. And so just kind of an example of what that actually looks like. Again, this is a scan from Q Recorder, seeing this color bias. So there was some work with the delta blue intensity metric to kind of see if, that would allow us to mathematically overcome this color bias. And in some cases it worked, in some cases not so much, especially within the Pseudotsuga genus. That was not actually super good. But then issues, of course, then with how do you deal with, treating the presence of non-mobile wood compounds, particularly resins, especially in the Southwest US. The Larix and Pinus especially are just incredibly chock full of resins, and kind of seeing, what pre-soak versus post-soak in ethanol or acetone, how those resin contents affected the subsequent data. So that was kind of an interesting exploration as well. And then, of course, resin ducts were an issue as well, particularly in the Pinus genus. We were looking a lot at white bark pine. This was problematic, especially for looking at the late wood. When your late wood cells are maybe, or your late wood ring is maybe only 2 or 3 cells wide, but there's a row of resin ducts that kind of just get in the way. Actually, to be honest, if anybody has any suggestions on how you get around that, I would love to chat about that more. Because yeah, in some cases I could kind of change the path that I drew in Q-Recorder around it, but ultimately sometimes it was impossible to get around. And this is also true for these rows of traumatic resin decks, especially in the Picea and Larix species. And a lot of these were oftentimes correlated with high insect outbreak years across the western US. And of course, as you know, with climate, temperatures rising, are highly correlated to kind of these increased severity insect outbreaks. So this is something that I'm sure, dealing a lot with more in the future, how to get around these when trying to measure blue intensity. As well as, so I worked primarily with Engelmann spruce, dealing with issues of fungal staining. This was kind of a big deal for Engelmann spruce, especially in the arid Southwest, in Utah, Arizona area, especially if there was propensity of beetle outbreak. This associated blue stain fungus in the outer portion of the ring. So I know that there's been work, I think it was Wang and others, we're looking at the delta blue intensity metric to kind of overcome this color bias from fungal staining or deteriorating wood. So that's something that I'm looking into as well. And so then, of course, deteriorating wood, this was another issue. So I won't really talk a lot about it today, but I was involved in a project doing a millennial length record. So combining living and remnant wood together. So how do you deal with these issues of when wood has been on the landscape for 1,000 years and it's deteriorated or discolored just due to weathering? So, and of course the answer was we did a lot of chemical treatments. And so this is a passive soak in ethanol in our fume hood here. So worked out pretty well. So just an example to kind of show you, this was a Douglas fir I was looking at. Pre-treatment, this was a passive soak in 95% ethanol for a few days. So what that looks like, beginning and then post-soak. It's not perfect, probably should leave it in longer, but the entire time we're taking measurements of kind of to see like how does the presence of resin affect the subsequent blue intensity data. And this was also true, as I mentioned, with that blue stain fungus, just trying to come up with different chemical treatments to deal with these issues and to see if it's reflected in the blue intensity data. And so this is an example from Engelmann spruce on the right with that blue stain fungus, and then it's a hydrogen peroxide based chemical treatment. So more on that in the future. And so really the meat of what I'm going to talk about today is the Engelmann spruce part of this network for my dissertation I developed. So I want to just start in the left-hand panel here. So this is a figure adapted from the N-trend network I showed at the very beginning, and kind of just situate this Engelmann spruce network that I was working with, these red dots here. And so over the course of 2 summers, I developed a network of 26 latewood blue intensity chronologies derived from Engelmann spruce. So these range northwards to northern Idaho, southwards to about, 120 miles north of the Mexico border in southern New Mexico. So the latitudinal gradient was pretty good. And again, targeting these locations below 45 degrees latitude primarily. And so once the latewood blue intensity chronologies were developed, my first goal was just to look at climate response. And so I ended up using the CREW 4.04 land data set to just kind of see what is the response of these chronologies and how do they relate to one another, as well as how do the latewood blue chronologies, how are they related to the tree ring width chronologies in terms of their temperature response. And so I looked at maximum temperatures as well as mean temperatures. And starting at the top of this figure here, again, it's just a correlation matrix based on a Pearson's correlation. I've got my months on the x-axis and my late wood blue intensity chronologies by site on the y-axis, BI on the top, ring width on the bottom. And so definitely we see maximum temperatures versus mean temperatures for the blue intensity. They tended to respond better to maximum temperatures overall. And that's pretty consistent from what I've seen a lot of Rob's work, right, in northern Canada as well. And then kind of secondly, where we see the strongest response is definitely August, this late summer months, which, you know, also kind of makes sense with it being the late wood blue intensity only. So then when we compare the late wood blue intensity versus the tree ring width, it's night and day, really. And this kind of gets at what I was saying earlier about, you know, that hydroclimate interaction with ring width historically. You know, it's not saying that ring width cannot be used for temperature reconstructions, you know, in the arid Southwest, but it's just, it becomes more complex. That signal is a little bit, you know, potentially more polluted by hydroclimate interactions. And so I also was able to test these relationships between the late wood blue and the temperature data to test for stability. And overall, I was very impressed. We didn't really see any divergence issues in the last 30 years or so. And so this was excellent. These chronologies, they went up to 2020, and so they're certainly up to date, which is great. And so kind of a little bit more about that ring width late wood blue relationship. So if I just ran a Pearson's correlation between the late wood blue chronologies and their tree ring width counterparts, a lot of times they're, you know, they weren't highly related, not really, or strongly related, but there were a few instances where they actually were. And so a lot of these were centered in Wyoming, in kind of these more mesic locations. And so it kind of got me thinking about, well, you know, maybe microtopography and site selection is going to be huge in terms of kind of looking at the differences in signal between these two metrics. So when I was able to kind of take those chronologies that were correlated with temperature, but also highly correlated with their late wood blue counterparts, and then I just ran a principal components analysis to kind of examine those signals further. And so the late wood blue intensity chronologies are in blue, the tree ring width ones are in red. And so if you just see PC loadings 1 and 2, kind of that distribution of where they fall, the late wood blue chronologies are most strongly loaded to PC1, whereas the tree ring width ones most strongly load to PC2. And so it's almost a slight bimodal or a bifurcated distribution here, which to me is saying, or I interpret it as, even these, you know, somewhat temperature sensitive tree ring width chronologies, there is still some pollution in their signal by some other factor, which I'm guessing based on the location and is probably precipitation. And so in terms of moving forward with this temperature sensitive tree ring chronology network, I just moved forward with the late wood blue intensity chronologies. And so, of course, my idea was to be able to create these regional reconstructions to kind of dissect regional temperature variability through time with this network of chronologies. And so, of course, if we look at, you know, panel B, revisiting this map from a few slides back, you know, there is spatial clustering in my sampling scheme. And of course, it's related to a couple different things, you know, the natural range, or the historical range, I guess, of Engelmann spruce, especially as we get kind of towards the southern range periphery, is kind of these island populations in a lot of cases, but also access to national forest and just site accessibility in general. So definitely plays into the geographic clustering. But just to kind of, you know, I guess corroborate that, I ended up running a principal components analysis again on just the late wood blue chronologies, and then doing a cluster analysis based on loadings PCs 1 through 3, just to see spatially how they're related. And actually, if you see where I decided to make the cut of dissimilarity, it fits really well with actually the geographic distribution. And so these are the spatial clusterings that I used for the candidate predictor pools for the subsequent reconstructions. And so once I had my chronology pools kind of spatially divided, I went ahead and compiled the instrumental temperature data, maximum temperature across these regions, and just did a climate response of each chronology to the regional chronology, or to the regional temperature data. And so what we see here is for the majority of regions, except the southernmost region, we start to see significant temperature response in April, and then it's pretty much done by September. And just like we saw with the local instrumental data a few slides back, the maximum response tends to be in August. And so calibration experiments between the predictor pools and the instrumental temperature data allowed me to define the optimum seasonality for reconstruction. And so, moving forward, it is important to note that the seasonality for these reconstructions, they do vary. And there's multiple reasons for that. We think about just using Engelmann spruce. A lot of it, I believe, has to do with the hybridization of Engelmann spruce kind of at the northern part of its range with other species of Picea versus, the southern range periphery and the southern Rocky Mountains for Engelmann spruce kind of influencing its climate response. So just something to keep in mind moving forward. And so I used a nested principal components regression technique to develop these reconstructions of the regional temperature variability. And so to kind of walk through this figure here, it'll translate through panels A through D. So starting with the northern Rocky Mountains, I've got my instrumental temperature data in black, and my reconstructed values in the colored lines. And so what you're seeing here is the relationship between the instrumental and the reconstructed values over the period 1920 to 2020. And then to the right, you'll see PC1 of those reconstructed, of the chronologies used in the reconstruction, the spatial nature of that relationship. And so ultimately, I was pretty happy with the response of these Latewood Blue chronologies in the reconstruction. Overall, pretty high R values, or Pearson correlation values translated as an R squared. We're looking at over 40% explained variance for all of these models for the common period. So I was pretty happy with that. And so being able to push these reconstructions back in time, right? So I used a nested approach. What that ultimately resulted in is being able to reconstruct temperatures back through time for the last several centuries. And so I've translated the reconstructed values into z-scores just for the sake of comparison between regions. And so these are 11-year averages just for reference. And so to break this down, if you think about anomalies, right? So z-score anomalies, anything greater than 0 is a warm year in red, and anything below 0 is indicative of cooler than average in blue. And so these are all scaled to the same mean and standard deviation. So again, think about it's different seasonality, so kind of something to think about when interpreting this figure. But really what I'm seeing is that we have pretty strong coherence at the multi-decadal to centennial scale in terms of trends, right? So the magnitude and duration of these historical warm and cool periods vary, but overall there is strong coherence, which we would expect, right? Just because of, the close spatial nature of this network. And so we're able to contextualize things such as the Dust Bowl, right? So these warm, dry events through time and kind of see how they compare to modern warming trends in terms of intensity, right? The modern warmth is certainly the most intense that we've felt across the Western U.S., at least over the past several hundred years. It also allows us to look at things like, the Dalton and Maunder minimum to kind of see, pulses, especially of the Dalton minimum, and kind of the intensity as well. So not only was I able to kind of use this study to look at, interregional variability, but also I use this as kind of an exercise in comparing Latewood blue intensity to, not only other Latewood blue intensity, but that MXD network that I talked about at the very beginning, the work by Briffa and others. And so kind of starting with that, you can see I've got my wiggle matching here on the top, but I want to mainly focus on this correlation matrix in panel C. So looking at the 4 records I developed with 3 independent but close proximity records. First starting with the BRIFA and others work, that MXD network. So they're pretty strongly correlated over the common or the shared period between all of these records, which again is not surprising given the geographic proximity, as well as the fact that, MXD and Latewood Blue, they tend to behave similarly in a lot of cases. And so, especially with the Central Rockies model being very strongly correlated to the BRIFA record, this was really good. But then also we're able to see the coherence of these records kind of with these larger northern hemispheric ensemble reconstructions. And so I wanted to start with the Guillet et others, 2017. It is a summertime reconstruction, so really there is also pretty good coherence here between my records and these records. And I think seasonality, of course, has a huge part to do with that. And then kind of one last thing for just this comparison slide, looking at these other more up-to-date records, such as the end trend records, I start to wonder about if one of the goals of this study was to increase the latitudinal representation southward of temperature proxy records. What does that mean for these hemispheric scale ensemble records moving forward? Could it be that, I know N-trend didn't include anything below 40 degrees latitude. Could it be that increasing representation latitudinally can improve these ensemble records moving forward? It's something that I think about and I think would be relatively easy to incorporate as, me and others, globally are, constantly pushing to look at, these high elevation locations further and further south. So I think there's a lot of promise there. And so kind of to conclude with this big chapter, this was like the 3rd chapter of my dissertation, I think the main takeaways for me is that, blue intensity, it is an effective tree ring parameter for reconstructing temperature, especially in these, new places, these, mid to low latitudes of North America, where before we just really haven't had a lot of work done with temperature because of constraints related to the parameter of radial growth. That being said, it is still in its infancy. There's a lot more to do. And as we recognize the current limitations, see them as opportunities to continually progress the science, whether it be pushing even further south, looking at different species, looking at how do we improve the common signal, how do we get clear images, better data, all things that have really come to light throughout this process for me. And then networks, of course, are important for characterizing these regional patterns of historical climate variability. And that oftentimes in these ensemble records, the interannual variability, it's muted. And so I think there really is a need for increased point density of these temperature sensitive proxies. And then, of course, improvements are still needed to the Northern Hemispheric record, as well as the Southern Hemispheric record. There's still a lot of work to be done. And, as a student doing my dissertation on this stuff, it was super exciting to be able to get to kind of have my little piece of the puzzle solved, or work towards solving it in terms of spatially improving this situation. And so then I'll kind of wrap up today with some current and future work. As you can imagine, I'm sure, when you've got a network of chronologies, there's just an amazing amount of data to work with, an amazing amount of questions moving forward to kind of, keep working, keep myself busy. So I wanted to start with a synthesis of blue intensity studies by genus, kind of situating the work I've been doing with the rest of the world right now. And so I even understand that I put this together, I think maybe 2 months ago, and this is already out of date. So blue intensity, our community is doing a really great things and we're doing things really fast. So there's studies that have come out since then that are not on this map. So I apologize for that. So what I've been kind of looking at, right, was predominantly the Picea genus, as well as the Tsuga genus. There's an east-west counterpart for this genus for North America. But I'm also really interested in looking at species that will improve the spatial gaps for North America, particularly in Mexico. The mountains of Mexico, we've got Douglas fir, that I'm interested in looking at. I'm working on a pilot study right now in southern New Mexico, just looking at, how feasible is it to actually work with this species that far south. I know that it works somewhat well in New Mexico. It's kind of a super music site, and I definitely know it works in Vancouver. So being able to look at new species to push this southwards, as well as if we see, the heartland of the United States, especially, where we're kind of in some places just devoid of trees, what are some other alternatives we can use to maybe bridge these spatial gaps? So right now I'm also looking at a population of Thuya occidentalis from western Wisconsin. So spatially, it's right about there, to kind of improve maybe these spatial gaps if, it's a new species that might be the answer. But I do want to just highlight the amazing amount of work that's been done just within the last 10 years by the community to increase the latitudinal extent. It's not just me, of course, in the Northern Hemisphere, especially as well. We're starting to get these Southern Hemispheric studies as well, which is super exciting to see. So kind of my biggest, I guess, project moving forward is this idea of developing a blue intensity network for North America, because I know that there are other people starting to, really get into these methodologies. So this image that I've got on the right is just kind of a example of a synthesis of data that I've collected over the past 3 years, or that I've processed. And so a huge part of this effort is not just, my dissertation collections, which are denoted by these blue X's in the Western US. But a huge part of this effort, and what I've spent a lot of time doing, is reaching out to other people to examine pre-existing collections. And so a lot of these are, of course, Ed Cook's from the East, a lot of his Eastern hemlock collections, being able to go back and revisit these collections that are in storage that, maybe were used for ecological studies, for, something completely different, unrelated to temperature, and seeing that if we reanalyze these collections and give them kind of sort of this new life, what that might result in. And so actually that's worked out really well for the Idaho lab of being able to create this network. It's like 136 latewood blue intensity chronologies that are very temperature responsive. And I know that there are other people doing it. And so I guess my hope with this is to foster collaboration moving forward across North America to kind of complete this network. And so what might we do with that? So I am currently working on a spatial field temperature reconstruction for the Western US. So once this network is complete, I foresee some sort of temperature atlas akin to, say, the North American Temperature Atlas, right? So kind of a taste of what's to come. And especially what is the utility of creating some sort of spatial field reconstruction. We can look at things such as, the North American Drought Atlas or the Seasonal Precipitation Atlas, and we can try to understand things like the role of temperature on drought through time. And so this is a schematic I'm putting together for a way that I would like to investigate that across the Western US moving forward. As well as another kind of fun project I'm working on right now is, I don't know if you all have heard, but the summer 2021 was crazy for the Pacific Northwest United States in terms of this heat dome that we had. And so contextualizing this, what they were calling a 1 in 1,000 year event with tree rings with blue intensity. So I'm in the process of, it's the first iteration of it, of putting together this reconstruction with latewood blue intensity, kind of contextualizing what 2021 actually looked like. And so based on it's a July-August mean temperature reconstruction. If we look at the relationship, it's really strong. It's, I believe, 70% explained variance is what I've got it to now over the instrumental record. But it's, yeah, just amazing in terms of unprecedented. So just some fun things I've been kind of looking at. As well as the idea with trying to develop this North American blue intensity network, looking at new species, right? So and using pre-existing networking groups to kind of get everybody speaking to one another. So so the longleaf tree ring network in the Southeast United States is a network that just launched. And so looking at, can we increase, our temperature records that far south, that far east for the U.S., where historically we don't have really comprehensive coverage there. As well as, of course, with you guys, with iBind. I think this is an amazing way to get everyone kind of on the same page and to network and collaborate. So I'm grateful for the chance to be able to share this presentation with you guys today. So with that, I will end it there.
Thank you very much, Karen. Brilliant talk. Quite a busy PhD.
Yeah.
So, well, I have lots of questions, but I guess it's not mine. I'll open it to the floor. People can put their, well, Mike Evans has his hand up already, so might as well start with Mike, if he's there.
I'm here. I'm just figuring out how to say hello. So first of all, could I just say that was a spectacularly fabulous talk, and I think they ought to not only give you a PhD, but a couple of postdocs as well for it. I have like 3 or 4 questions, but I'm just going to ask one because everyone else has to have a chance. I wanted to ask about going all the way back to like the basis of blue intensity. So it's thought of as a temperature indicator and a maximum temperature indicator, and you've shown those relationships. But going further back to your bringing up principle of limiting factors, I wonder if has this been tested? Have you been able to test the interpretation vis-a-vis principle of limiting factors in places where soil moisture might be a limiting factor, creating the anatomical features, which are then giving you the light features?
Absolutely.
And specifically, maybe you showed your transfer or regression analysis against maximum temperature and against temperature, but did you show that, for instance, against say, could you do that against precipitation, or precipitation that is the residual of the correlation with temperature? Thanks.
Yeah, absolutely. I totally could have included a slide on that. That was part of the screening process. So I just showed you, I guess, the nice looking chronologies that I included that when screened against precipitation, PDSI, temperature, min, max, mean temperature, the ones that made the cut, so to speak. So it was a network of, I think, about 40 chronologies originally. Yeah, and of course, a lot of them were from these arid locations that, although they were high elevation, over 3,000 meters, they were suffering the consequences of drought. And so, yes, those did not make it into what I showed today. But yeah, especially in the Sierra, that has been huge. I've been working with mountain hemlock there, and particularly, I've got a site in Yosemite that, for a good period of the instrumental record, probably until about 1980 or so, the response with temperature is very strong. But then post 1980, it's a signal shift to precipitation. So yeah, that's definitely happened, as well as with some of the Engelmann spruce at that southern range periphery in New Mexico. Those dry sites that certainly they've been screened against other climate variables, and they just didn't make the cut. And so, it's kind of, I guess I'm getting on an anecdote here, but it's kind of one of those things where with blue intensity this far south, thinking that you can just sample based on, for example, it's a north-facing slope. The soil seems like it's, holding moisture pretty well, and it's high elevation. And so I've checked all these boxes off the list, but then when you actually go run the data, that's not temperature responsive at all. It's soil moisture. So yes, that's been a huge, I won't say problem, but it's been an obstacle in some ways. So
yeah, I wonder if that's also an opportunity to get more information out of those data that have been screened out for the purpose of pure temperature reconstruction. Maybe they can be used for mixed temperature precipitation information retrieval.
Absolutely. Thank you.
Anyone else? I have a question. I mean, I noticed that Greg Wiles was sort of semi-online. I mean, you talked a lot about Tsuga, and we did quite a bit of blue work on Tsuga up in Gulf of Alaska, Delta Blue. I mean, it kind of worked, but it wasn't super strong. It wasn't as strong as Engelmann spruce. Any ideas why that could be the case? You know, or is it just a matter that we've got to measure maybe more trees and more sites to really get, you know, the R-squared values really high?
Right. Right. So it's kind of interesting. I feel like between the two of us, right, we've pretty much captured most of the Engelmann spruce's natural range. So, you know, the results that you've got at that range periphery versus what I've got at the southern range, yeah, I would agree that they're definitely, the spruce seems to be stronger. With the Tsuga, though, you know, I've had really pretty good results up until California. So I'm not sure. I do know that the latewood blue intensity for the Tsuga that I've sampled tends to work better than the delta blue intensity. So of course there's still that issue of color bias. So trying to figure out how to overcome that with maybe something other than the delta metric. But yeah, honestly, I don't know. I wonder if it's just sample depth potentially, but I have no idea, to be honest. I will say, though, I imagine, though, between Greg, you, and what Grant and I have put together, we probably also capture the mountain hemlock range pretty well throughout its range as well. So it would be interesting to, you know, do a latitudinal study potentially of looking at climate response with these two populations. That could be interesting. So.
Yeah, definitely speak to Greg. I mean, I think the Gulf of Alaska record is almost 2,000 years long now. So it's probably the longest blue record on the planet, I guess. I don't know. You know, so, you know, he's not here, he's hiding, but, you know, he should be writing up that paper, right? It's, but there, you know, and that record actually touches on this whole issue of mixing remnant material with living material and getting around or trying to get around that. And it's certainly, that is a challenge. That is a discussion for a later. Milos has a hand up. Oh, I don't know who came first. Christina is at the top of the list. So we'll go ladies first. Or is it Jesper? If it's Jesper, then go back to Milos.
I can speak for Christina then. Yeah, so very impressive talk, and I mean very impressive work. Really, we've done almost everything, it seems. But have you checked any signals in the early wood BI or anything like that?
Yeah, so with the Engelmann spruce in New Mexico, actually finding a really pretty strong spring temperature signal in the early wood. I decided to I've really only looked at the early wood for spruce just because of the color. With the Tsuga and some of the Pinus species, I'm just getting a little bit too much junk, I guess, color bias and just, I guess, resin ducts and other things. But the spruce actually, yeah, it's been pretty amenable to look at that. So.
Okay, but still temperature then, not.
Yeah.
Conduct
yeah, it was
signal or anything like that.
Yeah.
Milos?
Hi, Karen. Thanks so much for that super interesting talk. I really enjoyed it, and you know, clearly you've done loads of work and you know, a lot of work to sort of fill in that, you know, spatial gap in North America, and yeah, it's really nice to see this type of work. Actually, what I wanted to ask, Jesper kind of stole my question there with the early blue, so I'll scratch that one off, but I have a few other ones. So actually, kind of going back to the work you showed from the Appalachians, I was actually wondering about the relatively late temperature signal that you had there, September, October, with sort of the strongest response in October. It's kind of a little bit later than what we're typically used to seeing. So I was just wondering why, if you kind of have some idea why that might be the case. I would maybe guess, the relatively low latitude of the site and like a longer growing season, but I don't know the locations or the sites. So yeah, I was just wondering about that.
Totally. Yeah. No, I think those are probably wonderful explanations. It is the southern extent of red spruce at those locations. And then some of these places too that we were looking at, this Roan Mountain in particular. So this collection was actually, it was a pre-existing collection used for an ecological study. So there's part of me that's also wondering if it's, resultant from, partial, I guess, ecological factors with, acid rain with red spruce and kind of the dieback that is involved with that story. If that might be part of it, but I'm guessing it's probably just like you said, growing season, but also especially at that low of a latitude. It is a very warm, humid climate there. So, yeah.
Right.
It's certainly the latest I've seen across the network.
Yeah. Just kind of a related question. I also kind of noticed that the sites that you had there, sort of the signal strength, the temperature signal strength that you had was quite variable between the sites, even though they were kind of relatively, not too far away from each other. So I was just wondering if you have some idea why that might be the case.
Right. So some of those are red spruce, some of them are hemlock. One of them is a Carolina hemlock. So that was kind of, I think, partially part of it, right? It's like different species. But also I'm wondering too, within the Southern Appalachians, if microtopography is going to be more important there in terms of signal response.
Were there any big differences in elevation, or was it all kind of like close to tree line or?
Yeah, so unfortunately in the Appalachians, there's really no true tree line. So yeah, I'm wondering if because of that, it's like elevation really maybe matters more. One of those sites actually, come to think of it, was a cold air trap bog. The Alarka site was in southern North Carolina. So it's a climate refugia, so still you have the presence of red spruce, but it's not, at your 2,000 meters like some of the other sites were as well. That was kind of a proof of concept of, you think about places where even in the West we have these like spruce drainages, or we have spruce and we can't sample anywhere else, so heck, we might as well try it and see. So that's part of it as well.
All right, cool, thanks a lot.
Thanks.
Jack.
Yes, I'm totally new to blue intensity. My link to it is from having done perhaps a few studies on light rings, we're looking at specific rings rather than trying to get continuous chronology. And as Michael was saying too, when we're looking at light rings, there's light rings that are associated to drought and light rings that are associated to temperature. And that's what brings me to blue ring, is that we have, as you mentioned, a large collection of wood, and it's kind of interesting to kind of look at light rings in a continuous perspective. I had a question, and I don't want to give my age away, but I recall when I was in my PhD reading papers from Conke on red spruce in eastern North America, and she was associating maximum density, especially to spring temperature. And she was saying that through the red spruce decline, that signal was getting lost through time. So just out of just the questions, not to judge any of your work, I think it's fantastic, but just to see if you kind of look at perhaps the stability of the signal through times and things like that.
Yeah, certainly for that Roan Mountain paper, that was something we were concerned about. The cores that we were using for this study, they were, it was a pre-existing collection that was used to look at ecological succession, kind of following this high red spruce mortality event across the Southern Appalachians. Yeah, and so, we did not see it there, but I'm sure, in other places, that's probably very much the case. It is interesting to me, if we were to, I guess, sample across the broader range of red spruce, looking at was mortality of red spruce, was it worse at higher elevations versus lower elevations? And, what would a comparison of looking at, known mortality events in the Latewood Blue data versus the tree ringwidth data, if we see the presence in either of those series? Yeah, it's something I'll definitely have to look forward to the future.
Okay. Can I ask another question? Just from, I don't know if you can put the slide back. It went fast, but your reconstruction, your various regional reconstruction, it seems like you're doing very good with the high temperature, but not so good with the cool temperature. And
Right.
so I just was wondering what you may, if you can speculate on that. Or,
yeah, so certainly
the cool event, you were not doing very good, but if I'm not mistaken, it went very fast. So
yeah, no worries. No, I've noticed that across, especially, the southern, I guess the southern parts and the two southernmost reconstructions, that it's an overestimation in some ways of the cool temperatures. So, I'm wondering if that's related to temperature thresholds with, the chronologies I'm using at those sites. So, yeah, there's certainly room for improvement. That's for sure. So,
thank you.
Thanks, Joe. We're a bit over time, but I guess we're all spread around the world, so it's kind of meaningless maybe. Anyone? Well, I have a question just quickly. I mean, now you've done a very expansive network, and your predominant climate parameter, temperature parameter, is maximum temperatures rather than mean, which is something that we've consistently shown, in Western Canada all the way up into Alaska. Any ideas? And I know Mike's here, he's probably going to put his modeling hat on, but this is quite we don't really see the same thing in Eurasia, right? Mean temperatures is predominantly the dominant parameter. It calibrates better, and yet in northern North America, maximum temperatures. So is there a fundamental difference in the way the climatology or the data has been generated maybe, or are the trees just doing something a bit different? Any ideas?
Yeah, so I definitely, just from looking at the instrumental data, looking at the trends in maximum temperatures versus mean temperatures, especially over the last 40 years, especially, looking at spatial variation. So it seems like, the mean temperatures through and through, we're seeing, I guess more similarities in trends, whereas maximum temperatures across at least the Western US, that like really, I guess, strong spike isn't happening everywhere. And so I wonder if it's, yes, mean temperatures are increasing, but it's more about, minimum temperatures increasing potentially rather than maximum. So that's kind of, I guess, where I start thinking about that is, what are the trends in the instrumental data? However, I will say for the North American reconstruction, the spatial field reconstruction, I do think mean temperature might be the way to go. So that's what I'm working with now. Really, it's kind of like splitting hairs in some ways. Yes, maximum temperature is technically higher in terms of like a stronger relationship, but mean temperature is, I wouldn't say it's significantly less. You know what I mean? So.
Actually, actually related to that, with talking about the spatial aspect, and Kevin and grappled with this a little bit, is in the north end of your network, you're kind of having a broader season of, say, May, June, July, August. And then at the southern end of your network, you might have August, September. So when you're doing a spatial reconstruction, how do you get around the seasonal mismatch? Or are you going for a grid by grid flexible optimal season approach?
And unfortunately, no. The latter to the latter. So, yeah, just kind of looking at the calibration over multiple seasons spatially, where I see, the strongest red, I guess, propensity of dark red is kind of these calibration experiments across space, looking at, I guess, the proportion of gridded pixels that are above, 40% explained variance. That sort of thing, or above 50% explained variance. So, yeah.
Yeah, it's a tricky thing. Kevin and I debated discussed many times, and in the end we just went with the overall mean, which was the same, I think, as JJ. I can't remember. It was either May, June, July, August, or June, July, August. I couldn't remember, but it does have an impact with the spatial calibration, because you'll get this apparent failure in certain areas when actually it's the wrong season for the data that's in that location. And I'm not quite sure how to get around that.
You know, I wonder too if, you know, Dave Staley did the seasonal precipitation atlas, if that's a possibility of looking at, okay, well, if we have different iterations of this sort of gridded data product that's spring or just late summer or full growing season, or, you know what I mean? I don't know if that's realistic or useful. So, let's see.
Cool. Any other questions? Vishal.
So probably I'm not very original by saying that this was a fantastic presentation. And thank you very much for the network that you presented, and just more suggestion and maybe one more idea for your plenty of ideas that you already have, because with such a big network and also long chronologies, maybe it's worth to propose some collaboration with archaeologists, because you have excellent material, I mean, tool to date a lot of things which are probably not easy to date with ring width there.
Absolutely. Yeah, I think one of the major focuses is not only to improve the gaps spatially, but temporally. And we've got Rob's millennial length, my millennial length, and we've got one from Eastern Canada, I believe it's isotopes. But yeah, to be able to, you know, be able to go to these maybe unconventional sources of information that, again, bringing new life to these pre-existing data sets. I love that idea.
Andre, I think you have a question.
We're out of time, or what?
No, ask away. People can
okay,
leave.
Sorry, it's just a short question. Yes, thank you for this amazing presentation. Really, actually, I'm an archaeologist, so hello. And no, a question to maybe everyone, basically, because I didn't see that on your map. Is anyone aware if there's some blue intensity studies in juniper, tree-like species juniper?
Not that I know of.
Okay, thank you. Because there would be the issue as well with of softwood and hardwood color difference.
For sure. I mean, it depends on what juniper, but you know, if you're looking at very slow growing juniper, which is maybe often the case, then you're getting into potential resolution issues. You know, for some of
I mean, Excelsa, for example, though, and Fetidissima, those species that grow in Eastern Mediterranean predominantly, they have a rather regular tree growth. It's not like something shrubby, very very small and narrow.
So I know I was talking to Ed Cook about looking at blue intensity on Atlantic white cedar to be able to get one of these millennial length records for the East. We were brainstorming of ideas of how would you go about looking at, how do we get these long records? And, archaeological wood certainly is an excellent example. But if we think about, the long-lived and remnant material that we do have in the East, I think, juniperus is a really, could be promising. So, yeah.
I mean, there's loads of potential, and that's the nice thing about blue. I mean, blue has lots of problems, of course, but it's relatively quick and cheap, and you can assess species in a relatively short time. And that's and you don't need to measure hundreds of years, just measure out 100 years.
Exactly.
And then if it's got legs, then continue with it. I mean, that's really the first step. That's why it's nice. You don't have to go through all of that horrible sample prep that, Jesper looks like he's 50 now because he spent the last 10 years doing quantitative wood anatomy. It ages him. But there is advantage of quick and dirty sometimes. And I think if nothing else, certainly with you mentioned the Australasian stuff from New Zealand. I mean, that really was just more of a rough cut to see which species were worth the emphasis, right, for future work. You know, so yeah, go play with Juniper. You know, scan it, measure it, and see what comes out.
So
you know,
just another additional question. In the case where like you can't deal with the differences in softwood and hardwood color differences, then you go for the hardwood part. Like, would you include just one part of a sample, or what would you do? Like a rule of thumb. I know this is a very difficult question, but
yeah, no, I feel like to start, I mean, looking at that Delta Blue intensity to try it. I've seen people in the Western U.S. looking at pine that have a really intense heartwood sapwood that they just measure the sapwood. So
okay.
I don't know if that's
okay.
I don't know if that would get you enough data. Yeah.
I mean, it depends what your purposes are. If your purpose is dating only, then it's not an issue. If it's only really for climatology,
for climatology, yeah.
Yeah, then it's a problem. But I mean, you know, then that's, you know, Delta Blue seems to work on some species, and I don't think it's you know, that's another paper waiting to be written by someone. It's like, you know, when does Delta Blue work and why doesn't it work on some of the species? There's enough data out there probably to put that kind of synthesis together.
Have you noticed if like the Delta Blue works, for example, more on hardwood of a certain species than on the softwood or something like that? Or
no, it needs to counter. It is by design trying to counteract the color difference between the two, right? That's what the Delta Blue parameter is doing. So if you only measured the hardwood or only measured the sapwood, then you probably wouldn't need to do any correction. But you've got to be really careful bringing those data sets together. And then because you're splitting the data, then detrending becomes a bit of a headache as well because of segment length issues. So yeah, that's another story, you know, but that's, you know, in a way why we've put iBind together, because there are open questions. And hopefully as a community, you know, different people can focus on different aspects. You know, it's year one in the middle of COVID. We're not going to come up with all the answers, you know, and I think that's, you know, in 3 weeks' time when we had the next meeting and discuss about possible, you know, ideas for next year, we could actually have bespoke sessions on different aspects, you know, and getting a small group of people saying, okay, we're going to spend, I don't know, a couple of months measuring Delta Blue on different species, and then we come together for a comparison and see if, you know, there's all sorts of ways we can do this, you know, it's, yeah. So
thank you.
Yeah.
Thank you both.
Thanks. Yeah, Karen's talk, not me. My wife was here, she'll tell me I speak too much. So there you go. Anyway, any final questions before we call it a day or an evening, depending where you are?
Can I, one more?
Yes, certainly.
Okay, so you briefly talked about there was some species/sites that were correlated, where the ring width was correlated quite strongly with the latewood DI. Is that correlated with the average ring width itself? Or do you think that could be a resolution problem of those data sets, or is it just something that's, I mean, inherently there in the wood?
Right. So that's something, yeah, looking at, I guess, the narrow ring bias and some of the sites, I think may have had something to do with it, but ultimately, you know, looking at the ring width data across these sites, it's super variable. In a lot of these places, it's not, you know, super narrow rings equates to super low resolution blue intensity. So I don't think that was a major contributing factor. So the places where the ring width was most temperature sensitive, they were in Wyoming. They were kind of these more mesic high elevation sites that were Engelmann spruce. So I'm thinking it's just a natural, it's artifact of the climate sensitivity and maybe less about the resolution.
Mm-hmm.
But that is certainly something too that I could talk about that for days. Has been really frustrating to deal with, is when you've got these rings that are 2 cells wide, and okay, well, how am I supposed to get latewood, you know, and I haven't figured that out yet. So,
And there are people playing around with high resolution
yeah,
image capturing systems. So, are you thinking about that? Anything?
I don't think she heard your question.
What's it?
Yeah. I mean, there are people that are now playing around with high resolution image capturing systems like Mila and Dan Griffin, and here at BSL also. Are you thinking about getting something like that? Or?
I would hope to, yeah, and wherever I end up next. Yeah, so I've got our other PhD student at U of I, April. You may have met her when she was at WSL. So yeah, she's doing a comparison with resolution right now with her blue intensity data to see if the system that you all use, that high resolution, versus just the scanner, like the Epson scanner, to kind of see how that reflects in the data. So yeah, I would be interested in doing that myself as well.
I should say that Karen is finishing her PhD in the spring, isn't it?
Yeah.
So she's looking for jobs. So, you know, advertise, just helping you along there.
Thank you.
Right. I think, unless anyone, Jack, is that an old hand or a new hand?
That's probably an old one. I have a question, but they're more methodological and things like that. So I think I'm gonna. I know it's early here. It's only 11:30 in the morning, so I can go on for hours.
Well, I guess if anyone has further questions for Karen, I'm sure you can see it Heeter@uidaho.edu. So feel free to email her, or we can just arrange another meeting through iBind. You know, follow discussions in the future, right? So you know, we can't answer all the questions in one night.
Yeah, questions, and you know, if you're interested in, if anyone's interested in getting involved with kind of putting our heads together on getting this North American network together for this spatial field reconstruction, that's, you know, immediately what I'm working on now. So anything related to that, please feel free to reach out. Be great.
Brilliant. Well, thank you for everyone attending virtually. Maybe it will become more and more physical in the coming years. But so good luck everyone in Europe at the moment. It's a rocky road we have ahead of us, but we'll see what happens. Anyway, thank you everyone. Thank you, Karen, from everyone here.
Thank you.
Thanks.
And I'll send a link around for the next session on the 15th of December, and then we can all meet again. So on that, I'm going to go. Have a good evening, everyone, and afternoon. And see you all.
Thank you. Goodbye.
Thank you very much.
Good day, everyone.
Great. Thanks.
Bye-bye.
Thanks, Karen.